Hello students, today I am going to teach you all the first lesson of your grade 10 science textbook. As you know, the grade 10 science units have been identified as biology, chemistry and physics for you all, so that you get a better understanding of each lesson. The first lesson, Chemical Basis of Life, deals with living organisms. So, therefore, it is a biology lesson. So, in your environment, if you look at all the things around you all, you see a lot of organisms. Now, these organisms are animals, plants and microorganisms, including humans. So, we will look at how the bodies of these living organisms are made. Therefore, under this lesson, we will be looking at the following topics: carbohydrates, proteins, lipids, nucleic acids, water, minerals and vitamins. So, we will see how elements that make up the body of a living organism form compounds and exist in the body, as well as how it helps the living organism's functions. Chemical basis of life. The bodies of all organisms are composed of variety of chemical compounds. So, when we look at the environment, there are different types of organisms and the bodies of all these organisms have a basic component. Those are the chemical compounds. So, these chemical compounds are formed by the bonding of atoms of elements. Now, there are various elements and they can combine in different ratios, giving rise to many different types of compounds. If we look at these compounds, these chemical compounds are formed by the bonding of naturally existing elements in different ways. So, if we look at all the elements, there are about 115 elements in the environment. But out of those, 92 elements are the natural elements. These are the natural elements are 92. And out of these 92 natural elements, there are about 25 elements in the living body. Out of the 92 elements, about 25 elements are present in the bodies of living organisms. Out of these 25, carbon, hydrogen, oxygen and nitrogen, these four are the main elements. These are the main elements. These four main elements make up the bodies of living organisms. In addition to these main elements, there are other elements in smaller amounts. Those are sulfur, phosphorus, sodium, magnesium, potassium, calcium, iron and chlorine. We will be discussing these elements later: sulfur, phosphorus, sodium, magnesium, potassium, calcium, iron and chlorine. These elements also make up the body of living organisms. So, first we will see how much these elements are present in the body of a human being. So, here as you can see, this chart gives you the percentages of the four common elements present in human body. So, out of these, oxygen is the element that is present in very high percentage. There is 65% of oxygen in the human body, 18% of carbon, 10% of hydrogen and 3% of nitrogen. So, if we look at the elemental composition according to the mass in a human body, oxygen, carbon, hydrogen and nitrogen. The rest of the element make up only 4% of the human body. So, as you all can see, there are 4 main elements and there are other elements. All these elements combine together and form different types of chemical compounds. Now we will see what type of chemical compounds are there in a living body. So the chemical compounds are mainly grouped into 2 types based on the structure of these molecules. If we look at these types, one is organic compounds, the other one is inorganic compounds. Any compound that has carbon in it. Compounds that contain carbon are known as organic compounds. Compounds that contain carbon. Compounds that contain carbon are known as organic. Therefore, the compounds that do not contain carbon are inorganic compounds. Compounds that do not contain carbon. Now we say inorganic compounds do not contain carbon. But there are few exceptions. There are 3 types of compounds that contain carbon. So the exceptions are you have to remember this, oxides. When we say oxides, carbon dioxide, carbon monoxide, these are oxides of carbon and they are inorganic compounds. So, carbon dioxide, that is CO2, carbon monoxide, that is CO. These are the chemical formulae of these oxides. Then we have carbonates, that is another type. When we say carbonates, these substances contain the carbonate ion CO3 2-. So, any chemical compound that contains CO3 2- ions is called a carbonate. And the last one is bicarbonate. That is HCO3-. So, any chemical compound that contains HCO3-, that is bicarbonate ion, is called a bicarbonate. So, the oxides, carbonates and bicarbonates which contain carbon are all coming under inorganic compounds. So, these are exceptions, inorganic compounds that contain carbon. So, now we will see how these organic and inorganic compounds are present in the bodies of living matter and how they help us to function. Not only us, all the living organisms. So, if we look at the organic molecules, there are some organic compounds that are mainly present in organisms or living matter. Those are carbohydrates, proteins, lipids and nucleic acids. Because they are the main components in living organisms, we give them a special names. They are called biomolecules. organic compounds that build up the living body or living matter are known as biomolecules because they are present in the body of living organism, which is a biological system. We call it a biomolecule. So it has to be an organic compound and it is present in the body of the living organism. These four are the main types. And there is another organic compound which is the vitamin. You know about vitamins, the different types of vitamins and how you can obtain them. These are also organic compounds that are present in body of the living organisms. In addition to that, there are some inorganic molecules. Examples of these inorganic molecules are gases. When we say gases, the gas that we breathe in, that is the oxygen gas. So you all know, students, if we are breathing in oxygen, what do we exhale? Yes, we exhale carbon dioxide. Carbon dioxide. That is CO2. So these are the two main gases present in the living body. In addition to that, there is water, which is one of the main constituents in the living body. This is H2O. Water is H2O. So we have gases, water, and the third type is minerals. When we say minerals, those are the different types of elements which are there in different amounts and which are required for the functioning of the body of living organisms. So, as you all have seen, there are organic molecules. The main type is the biomolecule. Then we have the vitamins and the inorganic molecules which make up the living body of all organisms. Now, we will discuss where these elements are present in a human body. So, if we look at all the elements, as you remember, oxygen is in highest percentage, then we have carbon, hydrogen, nitrogen and the rest of the elements in small percentage. So, we will see where they are present, locations present in human body. If we take oxygen, that is 65%, it is present in all fluids. When we say fluid, any substance that can flow is known as fluid. So, as you all know, inside our human body, blood flows throughout the body and it carries a lot of substances along with it. So, this blood has oxygen in it. It is a fluid that has oxygen in it. Then if we take the human body, we have different tissues in our body. Skin is a certain type of tissue, the muscle is a certain type of tissue, then we have the bones, we have the nerves, all these are different types of tissues. All these tissues contain oxygen. Then the bones that give us a particular structure, that also contains oxygen. And finally, the protein molecules. So, oxygen is present in all fluids, the tissues, bones and in proteins. Then we look at the second element, that is carbon. Although it is only present in 18% of the body mass, it is present everywhere in the body. So, you take the full body of a human being, grown person or a child, it is there in all parts of your body. So, carbon is like the basic element present throughout the body. Then the third element is hydrogen. That again is present in all fluids, tissues, bones and proteins. So all these four components contain hydrogen as well. Then we have the fourth element, which is 3% by mass. This again is present in all fluids, tissues and proteins. So as I said, fluids like blood, then we have tissues, the different tissues like skin, muscle and etc. And the proteins in our body contain nitrogen as a constituent element. These are the four main elements. Then we have the other elements. Calcium is the first one, 1.5%. This is present in your brain, the lungs, part of the respiratory system, then kidneys, which is part of the excretory system, then the liver, which is part of the digestive system, the heart, that is part of the circulatory system. So here you can see the calcium element, although in small quantity, it is part of many different systems. And the thyroid gland, this is a gland present in the throat region of a human body and that produces thyroxine hormone. We will be discussing about hormones in detail later. These hormones are substances that are required to control the function of a human body. For example, the growth of the human body from the small days is controlled by a certain type of hormone. So, one of those hormones is secreted by thyroid gland. Now, this thyroid gland contains calcium. In addition to that, calcium is present in muscles and bones. Then we look at phosphorus. That is only 1% in the human body and it is present in urine. Urine means the bladder which stores the urine and also the bones. It is one of the constituent elements of the bone. Then we have potassium. Now this potassium is present as part of enzymes. Again, enzymes are protein substances which are needed to facilitate the biochemical reactions. Biochemical reaction means chemical reactions that take place inside the biological system, that is a cell or a human body or the body of a living organism. Now these enzymes contain potassium. Again sulfur 0.25%, this is present in proteins. Then we have sodium 0.15% and this is present in all fluids and tissues of a human body. Then magnesium, that is 0.05%, is present in again brain, lungs, kidneys, liver, heart, thyroid gland and muscles. Here you can see students, both calcium and magnesium are present in similar parts of the body, but calcium is present in bones, but magnesium is not present in bones. Then we have chlorine that is present in the skin cells. So all around your body, the skin is an organ that acts as a protective layer for the human body. Now this skin contains chlorine in it. The cells of the skin contain chlorine in them. Then we have iron. When we breathe air, we inhale oxygen and we exhale carbon dioxide. Now this oxygen has to be circulated throughout the body of a human being. So this circulatory function is done by a substance called hemoglobin. Now in this, the heme part means there is iron in it. So hemoglobin is a substance that is present in blood of humans, and this contains iron. And finally, we have iodine. Iodine is an element that is present in hormones of the thyroid gland. So students, I hope you all got a good understanding of the different types of elements, what percentage they are in the human body and where they are present. So now we will look at the different types of organic molecules that are present in the bodies of living organisms. So the first type is carbohydrates. Now when we say carbohydrates, this is the most abundant organic compound on earth. Most abundant. And they are produced by the photosynthesis process of green plants. Now when we say most abundant, it is there in many different organisms. So it is widespread and it is there in very high amount. And these carbohydrates are produced by green plants. Now you all already know, students, green plants in the presence of sunlight, they utilize carbon dioxide, water, and they produce their food. So when they use carbon dioxide and water in the presence of sunlight, for now we will take it as green plants. They actually need the component called chloroplast. So green plants in the presence of sunlight, they produce food. The food they produce is glucose. And in addition to that, oxygen gas is evolved. Now we are concerned with this glucose that is produced during photosynthesis. This is a carbohydrate. And this glucose can be converted to other forms of carbohydrates as well. So therefore, these carbohydrates become the most abundant substances that are present in living organisms. So from photosynthesis, carbohydrates are produced and they are present abundantly on earth. So if we look at food that contain carbohydrates or food which are rich in carbohydrates, we can take the different types of yams, for example, potatoes. Then there is sweet potato. These are examples of yams that contain lot of carbohydrate. Then we have the grains like rice, wheat, the different types of grains. Then there is sugar, flour. All these fruits, these are substances that contain carbohydrates. Now, as we see, carbohydrates are most abundant. Therefore, there are many different types of carbohydrates present. Now, we will see what elements are present in carbohydrates and how they can be classified. So, if we take the main elemental composition of carbohydrates, it is made up of carbon, hydrogen, and oxygen only. So, carbohydrates contain carbon, hydrogen, oxygen only, C, H and O. And out of these, always the hydrogen and oxygen are present in a fixed ratio. The ratio between hydrogen and oxygen in a carbohydrate molecule will always be 2:1. H2O, similar to that, 2:1. If there are two hydrogens, there will be only one oxygen. So, based on this, the carbon, hydrogen, oxygen are the elements present. Hydrogen and oxygen are present in 2:1 ratio. There is a common molecular formula for carbohydrates. As you all know, when we say a compound that has certain elements in certain amount. So, therefore, the way these elements combine is given by a molecular formula. This molecular formula gives the number of atoms of each element present in a compound. So, the common molecular formula of carbohydrates is CxH2OY. Now, we write this H2O in brackets to show that they have this 2:1 ratio, H2O. And here, X and Y are numerical values or numbers. They are numbers and they are whole numbers. Sometimes X and Y can be equal. So, if X and Y are equal, it is a certain type of carbohydrate. For example, if we take the molecular formula C6H12O6, in this X is C and H2O is also Y, that is also 6. So, X and Y are equal in this formula. But there are certain carbohydrates where the X and Y are not equal. So, X can be different from Y. For example, we take this carbohydrate where the molecular formula is C12H2O11. So, C12H2O11. Therefore, this has two different values for X and Y. This has the same value for X and Y. So, now we will see how we can classify carbohydrates based on their molecular formula and the structure of these carbohydrates. So, carbohydrates can be classified into three groups as below according to the way they are formed. So, under that we have three types: the monosaccharide, disaccharide and polysaccharide. Mono means one. So, there is only one sugar group or carbohydrate unit present in there. Saccharide means sugar. So, one sugar monosaccharide. The common formula, molecular formula of monosaccharide is C6H12O6. There are three different monosaccharides that we are going to discuss. All these monosaccharides have the same molecular formula C6H12O6. So, under this we will be looking at glucose, fructose and galactose. Glucose, fructose and galactose all have the same molecular formula C6H12O6 and they are all monosaccharide. Now, the second one is disaccharide. Di means two. So, two molecules of monosaccharides combine to form disaccharide molecule. We'll see how we can get the molecular formula for a disaccharide. If we take two molecules of monosaccharide, one is C6H12O6. The second molecule also has the same C6H12O6. So, when you add them together, it should have C12H24O12. But each time two monosaccharides combine together, there will be one water molecule removed. So, from this, there is one H2O molecule being removed. Therefore, the molecular formula of disaccharides become C12H22O11. We saw before also, in the common molecular formula, the monosaccharides have the same value for X and Y. But in disaccharides here, because there is a water molecule removed from combining of two monosaccharides, the values of X and Y are different. So, the common molecular formula of disaccharides will be C12H22O11. And under this, we will be looking at three types of disaccharides. The maltose, then we have sucrose and lactose. These are again formed by the combination of these three monosaccharides. And then we have the polysaccharides. Poly means many. So, many molecules of monosaccharides combine and form a large chain. Here, if we take each molecule as a monosaccharide, when each of these molecules combine, two monosaccharides combine, a water molecule is removed, minus. So, from this also there is a water molecule removed, minus. Likewise, each time two monosaccharides combine, a water molecule is removed. So, students, as I have explained already, when a polysaccharide molecule forms with the combining of many monosaccharide units, between two units of monosaccharides, one water molecule will be removed. Therefore, when we write the common formula, common chemical formula for polysaccharide, we will be writing it as C6H10O5. So, this will be the common molecular formula, C6H10O5. That is because between two monosaccharide molecules, a water molecule will be removed. So, we are removing two hydrogens and one oxygen from the monosaccharide unit. Like this, there are many polysaccharides combined together. Therefore, to denote that it is a polysaccharide containing many monosaccharide units, we use a subscript N that has to be outside the brackets. And as you can see here, N is the number of monosaccharide molecules present in a polysaccharide. As you already know, when these monosaccharides combine, they can combine in many different ways. Therefore, they can form different types of polysaccharide. As examples for polysaccharides, we will be discussing three types of compounds. One is cellulose, then we will discuss about starch and glycogen. All these three polysaccharides cellulose, starch and glycogen are made up of monosaccharide units. Because of their different combination, they will have different properties and different uses. As we have discussed now, these are the different types of carbohydrates based on their structure. So, under that, we will be discussing about monosaccharides, disaccharides and polysaccharides with their relevant examples. First, we will discuss monosaccharides. So, the first one will be the monosaccharides. Now, monosaccharide is the structural unit of carbohydrates. When we say structural unit, as we discussed before, these monosaccharides, when two of them combine, they form disaccharides. And many monosaccharides combine, they produce polysaccharides. So, therefore, they become the basic structural unit of carbohydrates. All the carbohydrates are made up of monosaccharide units. And the name simple sugars. They are commonly known as simple sugars. I said saccharide means sugar. So, because they are the simplest saccharides or simplest carbohydrates, we call them as simple sugars. They are crystal shaped. Crystal means when you feel it with your hand, you can feel that there are sharp edges to the solid particles. So, that is called a crystal shape or crystalline in shape. So, these monosaccharides are crystalline in nature or they are crystal shaped, and they generally have a sweet taste. You all have tasted glucose and you know that it has a sweet taste. So, monosaccharides have a sweet taste. And when you want to take glucose, you can either just put it into your mouth and just swallow it, or else you can dissolve it in water and drink it. This is something you would have done in your lifetime. So, here you can see that glucose is soluble in water. That is, monosaccharides are soluble in water. So, these are the common properties of monosaccharide. And as examples for monosaccharides, we will be looking at glucose, fructose and galactose. And as you already know, all these monosaccharides have the same molecular formula, C6H12O6. But the way these elements, the carbon, hydrogen and oxygen combine, they form different types of monosaccharides. And therefore, these monosaccharides have different properties and different uses. So, now we will see how they are useful. So, the first one is glucose, characters of different types of monosaccharides. Under that, we will look at glucose. That is the type of monosaccharide. We will see where it is present, the location it is present and the different facts, other facts about these molecules. So, the first one, glucose is present in ripened fruits. All of y'all would have eaten fruits and I am sure you love fruits. These fruits have a sweet taste and they contain glucose. So, fruits which are ripened, not in the raw state, only when they are ripe. Say for example, the mango which is yellow or reddish in color, when it is nicely ripe, then it will contain glucose. The second one is the bee honey. Honey that is obtained from bees. So, bee honey also contains glucose. Now, we will see some facts about glucose. Now, as you all know, I said before, during photosynthesis, the green plants produce glucose. So, that is how glucose is mainly produced during photosynthesis of plants. Photosynthesis of plants produce glucose. So, as I said, in the green plants, glucose is produced during photosynthesis process. Now, this glucose is converted to different forms of carbohydrates and it is present as storage food in plants. And when we eat food, that is the plant food, for example, rice or potatoes, they contain lot of starch. Now, this starch is undergoing hydrolysis during digestion within our body. As you know, students, when we looked at the common molecular formula of the different types of carbohydrates, you saw that when two molecules of monosaccharides combined, one molecule of water was released. At the same time, if you give or provide a molecule of water to a disaccharide, then it can be separated into monosaccharides again. So, this process where you give water, that is called hydro, and lysis means separation. So, by providing water, if you can separate a large molecule to simple molecules, then we call it hydrolysis. So, similarly, the starch, which is a polysaccharide, can be hydrolyzed inside the digestive system during the digestion process. And during this, glucose becomes the final end product. So, we say glucose is the end product of starch hydrolysis. It is the end product of starch hydrolysis. So, when this glucose is produced within the body, that is after digestion, that gets absorbed to the bloodstream. So, glucose gets absorbed to the bloodstream. Absorbed into blood. And the blood transports the glucose throughout the body and the glucose is distributed to all parts of the body. And whenever the cells of the body need energy, they take this glucose, they utilize this glucose and break it down to form energy, produce energy. This process in which glucose is broken down within the living cell and energy is produced, it is called cellular respiration. So, glucose is broken down in cellular respiration to produce energy. To produce energy. So, as you can see, glucose is produced during photosynthesis in the green plants. Then it gets converted to other forms of carbohydrates. It's consumed by animals, then undergoes digestion process where during starch hydrolysis, glucose is the end product. This glucose is absorbed into blood, transported throughout the body and in cellular respiration, it is broken down to produce energy. This energy is utilized by the body of living organisms. So, these are the locations of glucose and the effects of glucose. And here you can see the structure of a glucose molecule. Remember students, you will not be required to write the structure for your exam. But if you see the structure, you should know to identify it. If you remember the molecular formula of glucose, C6H12O6. Now you can look at this structure. There is one carbon on top, then all these points. Those points of the hexagonal structure are carbon atoms. So we have 1, 2, 3, 4, 5 and 6 carbon atoms, 12 hydrogen atoms and 6 oxygen atoms in this whole molecule. So this is the structure of a glucose molecule. As you all know, all the monosaccharides have the same molecular formula. This is the structure of glucose, but the structure of fructose and galactose will be slightly different. That is why they have different properties than glucose. So now we will see where they are found in a living organism and what their importance is. So the next or the second monosaccharide is fructose. From the name fruct, we can assume that it is related to fruits. So fructose is one of the main components in ripened fruits. A sugar found in ripened fruits. Therefore, we call it the fruit sugar. Also, it is present in bee honey, pumpkin and carrots. And I'm sure you would have eaten all these food items and you know all of them have a sweet taste. So fructose is the sugar that has the sweetest taste. So this is the sweetest sugar. It is known as fruit sugar. This is the sweetest sugar and this is formed during the ripening of fruits. It is formed during the ripening of fruits. So the fruit sugar, it has the sweetest taste and it is formed during the ripening of fruits. Those are the importance of fructose. Then we will look at the third type of monosaccharide, which is galactose. This also has the same molecular formula, C6H12O6. This is mainly present in dairy products. Dairy products means products made from milk. So the reason is there is a substance called lactose, which is also a carbohydrate and you all know it is a disaccharide. So lactose is made up of galactose. It has one monosaccharide, that is galactose. So when lactose is hydrolyzed, galactose is produced as the product. So galactose is the product of lactose hydrolysis. And also you all know that milk does not have much sweet taste. So that means this monosaccharide galactose also has no sweet taste. So these are the properties or importance of galactose. I am sure students, now you would have got a very good understanding about how monosaccharides are formed by the combination of the basic elements and also what their types are and their importance is. So now we will look at the structure and uses of disaccharides. So disaccharides are formed when two monosaccharides join together. So two monosaccharides join to form a disaccharide molecule. And during this process, a water molecule is released. And in the same way, relevant monosaccharides can be obtained by hydrolyzing disaccharides. So, if we put this into a formula, you can see that one monosaccharide combines with another monosaccharide, which produces a disaccharide. Di means two sugar molecules, and a water molecule is released during this process. Two monosaccharides, two molecules combine and form a disaccharide, releasing water. If we write the same thing in a chemical formula manner, as a chemical equation, we will be showing it as C6H12O6, a monosaccharide, another C6H12O6, the second monosaccharide, giving C12, two additions of the carbon, but H22 and O11, because a water molecule is removed from the two molecules of monosaccharide. So, in this process, the two monosaccharide molecules combine and form a disaccharide, releasing a molecule of water. At the same time, when you provide water to a disaccharide, it will get hydrolyzed and produce the monosaccharide back. So, therefore, we can show this as a double equation. The forward reaction is where disaccharide is formed, releasing water, and backward it is the hydrolysis of disaccharide. The disaccharides again have some properties. They are also sweet in taste. They are sweet. Disaccharides have a sweet taste. And also they are water-soluble crystals. So, as we said, we can dissolve disaccharides in water. So, they are water-soluble and also they have a crystalline shape like the monosaccharides. And the examples for disaccharides are maltose, sucrose and lactose. So, now we will see the locations and the uses of different types of disaccharides. The characters of different types of disaccharides. Under that, first we will look at maltose. Now, maltose, from the word malt, it is present in germinating seeds. It is present in germinating seeds. And one molecule of maltose is made from 2 molecules of glucose. It is made up of 2 molecules of glucose. If we put this into an equation, 2 molecules of glucose forms 1 molecule of maltose, releasing 1 molecule of water. And also, this is an intermediate product of starch hydrolysis. Now, this you would have experienced if you eat a little bit of cooked rice, only rice without any curries. If you take a little bit into your mouth and if you keep on chewing that rice, you will get a slight sweet taste. Now, this sweet taste is because the starch present in rice is hydrolyzed within your mouth and maltose is produced as the intermediate product. So, maltose is an intermediate product of starch hydrolysis. So, the next property is that maltose is an intermediate product of starch hydrolysis. Maltose is made up of 2 glucose molecules. Therefore, it has a molecular formula C12H22O11, and during the production, water is released. At the same time, when starch is hydrolyzed, maltose is produced as an intermediate product. Now, we will look at the second type of disaccharide, that is sucrose. Now, sucrose is the carbohydrate or disaccharide that is present in sugar, the sugar that we use at home. So, sucrose is a component of white and brown sugar. It is present in sugar cane. It is present in beet, in some fruits, it is present in the phloem sap of plants. It is present in white and brown sugar, sugarcane, beetroot, some fruits and phloem sap. You all would have tasted all these substances. We will see what phloem sap is. Now, as we discussed before, in green plants, in the leaves, there is photosynthesis taking place. And as the end product of photosynthesis, glucose is produced. But in the form of glucose, it cannot be taken all around or it cannot be transported to different parts of the plant. So, for that purpose, glucose is converted to sucrose, and this sucrose is translocated. The name of that process is called translocation of food in plants. During this translocation of food in plants, sucrose dissolves in water, and this composition is called the sap of plants, and this sap is translocated through a tissue called the phloem. So in the stem of the plants, there are tissues that are there to transport substances. And the tissue that is present to translocate food is called phloem. And the process is called translocation. And the component that is translocated is called the sap. Now we will see what this sucrose molecule is. As you know, all disaccharides are made up of monosaccharides. The sucrose molecule is made up of one glucose molecule and a fructose molecule. So when a glucose and fructose combine together, they form a molecule of sucrose and release a molecule of water. So sucrose is formed from glucose and fructose. And all these substances, the food substances, are very sweet. And sugar is one of the sweetest substance. So that means the sucrose disaccharide also has a sweet taste. This has a sweet taste. So as you can see, these are the places where you get sucrose and the main component of translocation in plants. Therefore, it is present in sap of phloem, and also it is made from glucose and fructose, and it has a sweet taste. So that is the second type of disaccharide. Now we will look at the third type of disaccharide, that is lactose. So lactose again is present in dairy products. Products made from milk. Dairy products. Products that are made from milk. Now the lactose molecule again is made up of two monosaccharides. One is glucose. The other one is galactose. So when a molecule of glucose and galactose combine together, you get a molecule of lactose with the releasing of water molecule. Now this lactose is one of the main components of milk. And as you know, milk is not that sweet as sugar. So that means lactose is not so sweet as sucrose. So here we can say not sweet as sucrose. And this is the main component or compound present in milk. And there is about 4 to 6% of lactose in cow's milk. And 6 to 7% of lactose in human milk. So when organisms drink milk from their mother, they get this lactose. So that in turn is made up of glucose and galactose. So that gives again energy to the living organisms. As you all see, lactose is the main component in milk. So milk is produced in animals. Therefore, lactose is the only carbohydrate that is absent in plants. Only carbohydrate absent in plants. these are the facts of lactose, a disaccharide. Now we will look at the different types of polysaccharides. So polysaccharides, from the name you can understand poly means many. Many monosaccharides combine together. So a polymerization of a large number of monosaccharides form a polysaccharide molecule. Remember students, I explained before, when two molecules of monosaccharides combine, one molecule of water is removed. So here many molecules of monosaccharides combine together. Therefore, lot of water molecules will be removed and a long chain of polysaccharide will be formed. Therefore, it has a common molecular formula C6H10O5, and with we write brackets, and out of the brackets we use the letter simple N to denote that there are many monosaccharide units present in the polysaccharide molecule. Now, so that process by which polysaccharide is formed is called polymerization. As we discussed before, when monosaccharides combine, water is released. At the same time, when you provide water to the relevant polysaccharide, it produces the relevant monosaccharide. So, monosaccharide combine and form polysaccharides. Polysaccharides are hydrolyzed to form monosaccharides again. And since these are very large molecules, they are polymers. Polymers means very large molecules. They are not soluble in water. They are insoluble in normal water. You cannot dissolve these polysaccharides easily in normal water. And they are not crystals. They are not crystals. If you remember, the monosaccharides and disaccharides were soluble and they were crystals, but the polysaccharides are insoluble in normal water and they are not crystal shaped. So, the structural unit of polysaccharides is glucose, and these glucose molecules combine in very large number and in different forms, giving rise to different types of polysaccharides. When many glucose units combine in different forms, we get different types of polysaccharides, and the examples are cellulose, starch and glycogen. These are the three types of polysaccharides that we will be discussing about. Now, we will look at the first type of polysaccharide. It is cellulose. Now, this cellulose is a polysaccharide which is present in the cell wall of plant cells. It is present in cell wall of plant cells. Students, if you remember, you know that all the organisms are made up of cells. All their living bodies consist of cells. In all these cells, there is a boundary line for the cell, which is called the cell membrane. This is a protective layer for the cells. But in addition to that, in plant cells, now if you look at this cell, this is a plant cell. A plant cell has this dark green color layer. You can see it is a cell wall. So, in plant cells, there is a cell wall, and this cell wall is made up of cellulose. So that is where you get cellulose. The second one is in fibers. So we get cellulose in the cell wall of plant cells and in fibers. That is also in plants. Now here you can see in a plant body there are fibers in between. If we look at the magnified region of a plant stem, a young plant, from this the stem is magnified, and in the stem if you look at the tissues are made up of long thin structures which are there for strength and other purposes. These structures are called fibers. These fibers contain cellulose. So the cell wall of plant cells and fibers contain cellulose. When we eat food that is rich in cellulose or that contains cellulose, this cellulose does not digest within the digestive system. So here cellulose does not digest in the human digestive system. Therefore, cellulose cannot be absorbed by the human body and it does not have a nutritional value, but the importance is cellulose helps to prevent or avoid constipation because it helps to give out feces from the body. It helps to avoid cellulose helps to avoid constipation because the fibers help the removal of feces from the body. Feces, you all know, is the undigested food which is there in the human digestive system. When you eat food, most of it digests. That undigested component in addition with a few more substances is called the feces. This is not an excretory product. It is only the undigested food. So this should be given out or removed from the body. That is facilitated by cellulose. Therefore, it helps to avoid constipation. Now, we will look at the second type of polysaccharide, that is the starch. Now, starch again is present in many food items like yams. When we say yams, potato, sweet potato, all these have starch. Then cereals. Cereals contain starch, grains contain starch. So these are some substances that contain starch. Yams, cereals and grains. When we say grains, rice, wheat, these are flour, that also contains starch. Contains starch. So this starch is the main storage component present in plants. The carbohydrates are stored in the form of starch in plants. Carbohydrates are stored in the form of starch in plants. So students, as I said, carbohydrates are stored in the form of starch in plants. And these are some of the food that contain starch. In addition to these, the yam, cereals, grains and flour, we can also consider jackfruit and breadfruit, which are rich in starch. So these are the plant products that contain lot of starch because it is one of the forms in which carbohydrates are stored in plants, the main form in which carbohydrates are stored in plants. Now we will look at the last type of polysaccharide, that is glycogen. This is present in animal liver and muscles. In animal liver and muscles. Like starch, glycogen is the main storage component or the type in which carbohydrates are stored in animals. So we can say carbohydrates are stored in the form of glycogen in animals. So out of the three polysaccharides, starch and glycogen are main storage components in plants and animals respectively. So that brings the discussion to the end of the different types of carbohydrates and their importance in living organisms. Now we will again summarize the significance of carbohydrates for living organisms. So as I said, significance of carbohydrates, it is an energy source. So under this we said glucose is the main component that is broken down during cellular respiration. So therefore carbohydrates are an energy source and the cellular respiration produces and provides energy to the function of all the cells in the body of living organisms. The second example or the second significance is as a storage compound. So as we saw before, both the starch and glycogen are two types of carbohydrates which are main storage components in plants and animals. Starch in plant and glycogen in animal. So as a storage component. Then the carbohydrates are present as structural components in the plant cell wall. So that again is a polysaccharide. As you know, it is cellulose that is present as the structural component in the plant cell wall. And finally, it is a constituent of nucleic acid. Now, we have not discussed about nucleic acid yet. They are also a type of biomolecules, and in these biomolecules, there are carbohydrates which are pentose sugars. The nucleic acid contains pentose sugar. This is a monosaccharide that is present in nucleic acid. Okay, so as we come to the end of the discussion, I hope it is clear to you how carbohydrates are formed, the different types of carbohydrates and the significance of carbohydrates. And as you know, different food items contain different types of carbohydrates. So now we will see how we can use different types of tests to identify the presence of these carbohydrates in different types of food. Test to identify carbohydrates, and under that first test is the starch test. So as you know, there are many carbohydrate containing food, and out of that, the food like potatoes, rice, they contain high amount of starch. So for this activity, we will be using potato as the food substance that is tested for the presence of starch. So here we take a potato. So these are the other materials that are needed to carry out this activity. I will label them as I explain the procedure. So first we have to take the potato, we have to clean it, we have to remove the skin, cut it into small pieces. Then we have to grind the potato using a little bit of water and make it into a paste. And from that we can get the starch solution. So to do that, we take the cleanly cut pieces of potato into this laboratory equipment, the mortar and the pestle. Normally in the lab, we use the mortar and the pestle to grind substances. So this is the mortar and pestle. So we take the pieces of potato into the mortar, add a little amount of water, grind it well. Once it gets grounded, you can add a little bit more water, dissolve the substance, and we can decant, that is just pour the solution only into a test tube. So decant the solution into the test tube. So to do the activity, we will need the test tube as well. We have all the necessary substances. We have the potato, the food containing starch, the mortar and pestle to grind the potato. When grinding, we will need a little amount of water. So for that, we have water in a beaker. That is used to grind the potato. So after doing that, we take the potato solution, or what we need, the starch solution, into the test tube. And this is the chemical substance. We call it the reagent that is needed to test for the presence of starch. This is called iodine solution. It is a liquid iodine solution. So now we have the starch solution in the test tube. We add a few drops of the reddish brown color iodine into the solution. And then you will see the color change clearly. So students, you have understood the activity that is carried out to test the presence of starch in food samples. Now I will write down the steps and summarize it again. So if we write down the methods, if you are asked to write down the method of carrying out the test for starch, then you will have to start by saying, first we need to prepare a solution of starch. This is done by grinding the starch containing food using a mortar and a pestle. So first we take the food, add a little bit of water, then grind it nicely using a mortar and pestle. Then after we have prepared the solution, we decant, that is to remove the solid particles and to take only the solution. You don't have to really go through the process of filtering it. Just if you pour the solution from the mortar into the test tube, that should be sufficient. So decant the starch solution into the test tube. So we can take this starch solution that we have prepared from a piece of potato. At the same time, we can straight away use a piece of potato. As I told you all before, we clean the potato, we take the potato and without grinding it and making it into a solution, we can cut a slice of potato and use it as a sample containing starch. In addition to that, students, you all know there are many different food items that contain starch. For example, the bread we eat, that is made up of wheat flour. So we can use wheat flour as a starch containing substance. String hoppers that we eat, usually the white color string hoppers, because that is also made up of wheat flour. So for this activity, in addition to potato, which is a starch containing food, we will be using a piece of string hopper and a small amount of wheat flour. I will include that in this step. So in addition to this, in addition to the potato extract, string hopper, wheat flour, and a slice of potato can be used. As the starch containing material. So now we have the food sample ready. Then what is the next step? We have to add a few drops of iodine solution to the food sample. So the next step, add a few drops of iodine solution to the food sample. Then you will get the required observation. So students, you can see this is a very simple activity. Either you can take a potato, grind it using a mortar and pestle and get the extract of starch, or else you can use a piece of potato as the starch containing substance. In addition to that, we can use any food that contains lot of starch. And the reagent that we are going to use is iodine. Now that you have understood this activity clearly, let us carry out this activity in the lab. Okay students, now we will do the activity to test for the presence of starch in biomolecules. As food samples, I have 3 samples here. A piece of string hopper made from flour, a piece of potato and flour for starch. Now you all know we need to use one particular reagent, that is a solution of iodine, which is reddish brown in color. The procedure is to add few drops of iodine to the food sample and observe. Before I start the activity, I will also add a small sample of flour onto the tile. Here we are using a white tile, so you can see the color changes also clearly. Now I draw a drop of iodine and add it to the string hopper. Another drop of the iodine solution to the piece of potato and another drop to the flour. You can see the color change, the appearance of purplish blue color or dark purple color in the piece of string hopper as well as flour. But the color is not very visible in the piece of potato. So I will add another few drops of iodine. Here you can see slightly the purple color appearing on the piece of potato. If you look at the center of the potato, you can see the color change. So students, you can observe all the 3 samples. Observe clearly the color that has appeared, a very dark purple color. This confirms the presence of starch in the food sample. So when you use iodine solution, the reddish brown color of the iodine solution should become a purple color. That will confirm the presence of starch. To show the difference between the normal color of iodine as well as the color change with starch, I will put a few drops of iodine on the tile. You can see students, a light reddish brown color at the same time. With the 3 samples, you can observe the purple color. So you can see the color change very clearly. Okay students, you would have properly watched how we did the test for starch and then you would have seen the observation. You saw clearly the color change. Now recollect the color change. The reddish brown color of iodine became a dark blue purple or blackish purple color. So the observation is reddish brown color of iodine becomes dark blue or purple. That will be the color change. The reddish brown color turns to dark blue or purple once you add the iodine solution into the starch solution. So from this we can conclude that there is starch in the food sample tested. So that is the conclusion. the appearance of dark blue or purple, or sometimes it is referred to as blackish blue color because it's a very dark color. This color confirms the presence of starch in the food. So that is the first test we do for carbohydrates. Now we will look at the second type of test that we will have to do. This is the test for glucose. As you know, glucose is a monosaccharide. It has a crystalline structure and it is soluble in water. These are the materials that we need to do this test. We need a sample of glucose. You have seen this glucose and you can notice that it is the crystalline white color solid. And the reagent that is necessary to test the glucose substance is known as Benedict's solution. Benedict's solution. In this activity, we will have to use the Bunsen burner. So, to keep a vessel on the Bunsen burner and heat it, we will be using a tripod and a wire mesh. So, as you all know, we can directly heat a reaction mixture using a boiling tube, or else we use a tripod and a wire mesh. We place it on top of the flame of the Bunsen burner and we can keep something like a beaker, a glass vessel on it and heat it. So, here we will be using a beaker with water in it, and this is given a special name. It is called the water bath. The purpose of a water bath is to heat other reaction materials without providing direct heat. So, what we do is we place the beaker with water on the tripod. That will become hot, and into that we place this test tube or the boiling tube containing the glucose and the Benedict's solution. Then that will become hot. This is to make sure that we see the observation clearly. So, here we use water bath. Water bath is water contained in a beaker, and also we use the test tube to carry out the reaction. So, this is how we are going to do the activity. We will have to take a small sample of glucose, few teaspoons of glucose into a small glass beaker, and as you all know students, we will be using, in addition to these main materials, we will be using other materials like small beakers, pair of tongues, which are available in the laboratory for this activity. So, we will take a small amount of glucose into a small beaker, then we add a little bit of water and dissolve it using a glass rod. Then after that, we will transfer a part of the glucose solution into this test tube. And into that test tube, we will be adding few drops of Benedict's solution. And once we add that, we place the test tube. Before that, we will have to set the tripod to the Bunsen burner, and the Bunsen burner, the flame will be burning, and on that we place the water bath, the beaker with water, and into that we place this test tube. And then you will see the observation. Now, I will again summarize the procedure of carrying out this test for glucose. So, if you are asked to write the method of carrying out the test for glucose, first you will have to prepare a solution of glucose. Then secondly, we will take a small amount of glucose solution into the test tube. Then to this, add a few drops of Benedict's solution. Now, as you saw, the Benedict solution is blue in color. Then what we do, use a water bath and heat the reaction mixture. So, as I said, to heat the reaction mixture, we will be placing the beaker containing water on the tripod on top of the Bunsen burner, and that will become hot. The water inside will become hot and the heat will be transferred to this test tube. We can't be holding the test tube throughout the reaction. So, therefore, what we do is we use a stand to fix the test tube. So, I will add that also in the materials required. We will need a stand to fix the test tube. Okay. So, we have the mixture of glucose and Benedict solution inside the water bath. Then you will get the required observation. I hope you clearly understood the steps involved in the test for glucose. So, now we will do this activity. Okay students, now we will do the activity to test the presence of glucose in food substances. For that, we will need glucose. To dissolve glucose, water. Then the reagent that we are going to use is Benedict solution. You can see it has a nice blue color, and that will give a color change with the glucose solution. So, to prepare the glucose solution, I will need a boiling tube. We will be using a boiling tube because we have to heat the solution. First, I will prepare a solution of glucose in a boiling tube. We will need a boiling tube because we have to heat the mixture. So, to that, I will add certain amount of glucose into the boiling tube. And add water. As you know, since glucose is a monosaccharide, it can dissolve very easily in water. And we make sure that it dissolves well. Now that the glucose is dissolved and we have the glucose solution, to this I will add a little amount of Benedict solution. When we add a small amount of Benedict solution, you can see it has a nice blue color. But the observation of the activity will be after heating the solution. We can directly heat this solution in the flame, but that will give rise to a very fast color change and you will not be able to observe it clearly. In order to avoid that, we use a water bath. A water bath is a beaker containing water and we heat the water. So gradually the heat from water will be transferred to the solution mixture. To place the boiling tube inside the beaker so that it will become hot gradually. Now you can observe the color change clearly. You can see at the bottom of the boiling tube there is a light green color appearing. So the mixture will completely become green in color first. After green, you can see the appearance of yellow color. I will mix the solution slightly so that the color is uniformly distributed. The yellow will now turn to a brick red color, and finally there will be a brick red precipitate appearing. You all can see the solution has turned from yellow, it has turned almost to a reddish color. You can see, students, the appearance of a brick red color. Now you can see the color change clearly. The solution has become brick red in color and the precipitate will deposit at the bottom of the solution. So students, you have observed the color change clearly. The blue color of the copper sulfate became green, then it became yellow-green, finally it became a brick red color precipitate. So this color change confirms the presence of glucose in food samples. Okay students, now you had a good look at the activity we just carried out in the laboratory to test for the presence of glucose. And you saw the nice clear color change. That was possible because we heated the solution using a water bath. If we heated the solution mixture directly in the flame, the color change would have been too fast. So you would not have been able to observe it clearly. But now you observed it very clearly, and the color change started from the blue color of the Benedict solution. Then this blue color became green and that turned to yellow-green. This in turn became orange. And finally, it gave a brick red precipitate. A brick red precipitate. So you observe the color change clearly and you can remember the blue color becoming green, then becoming yellow-green, orange, and finally the appearance of brick red precipitate. So this allows us to conclude that the substance we tested is glucose. So the conclusion of this activity will be the sample contains glucose. So when you see this color change with Benedict solution, you can confirm that glucose was present in that sample. So now we will do the next test, that is done to test the presence of carbohydrates. This is the test for sucrose. As you know, sucrose is a disaccharide which is made up of a molecule of glucose and fructose. So these are the materials that we need to carry out this test, and again we use the Benedict's solution. Benedict's solution as the reagent to test the presence of sucrose. Then for the sucrose we take sugar. Sugar is one of the food items that contains sucrose. Then we need to use a test tube. We will be needing more than one test tubes, and again, as in the previous activity, we will need a water bath to heat the solution or the reaction mixture. A water bath is a beaker containing water, and then we need a tripod and a wire mesh to place the water bath on when heating, and we need the Bunsen burner to provide the heat. This is the common burner used in the laboratory, and also we need an additional reagent, that is the sulfuric acid. We will be using dilute sulfuric acid. And in some instances, if we need to neutralize the excess sulfuric acid, we might have to use sodium carbonate solution. Sodium carbonate or sodium bicarbonate solution. This is not a must, but in activities we can also use sodium bicarbonate solution to neutralize the excess sulfuric acid. So this will be the procedure of the activity. First, we will have to take a small amount of sugar into a beaker. It can be a small beaker. Then we add a little bit of water and dissolve the sugar well using a glass rod. Then what we do is we take a small amount of that solution into the test tube. To that we add Benedict's solution. A few drops of Benedict solution, and like we do for glucose, we place this test tube inside a water bath, place the water bath on the tripod, and to hold the test tube for a long time, we also use a stand. A stand to hold the test tube inside the water bath until it gets heated. Then we use the Bunsen burner to provide the heat. This is the first part of the activity. Once you do this, you will not get any change. There will be no color change. I will first write down these steps and then explain the second step of the activity. Second part of the activity. So in the first part, we produce a solution of sucrose, solution of sucrose, that is sugar, is prepared, then we take a small amount of the solution into a test tube. We add few drops of Benedict's solution and heat the mixture using a water bath. This is the first part of the activity. Here we will not see any color change as we expected. So then we do the second part of this activity. I will go back to the previous slide. Here what we do is we take another test tube. Into that we again take a freshly prepared sugar solution. So you can either dissolve the sugar again in water and prepare a fresh solution of sucrose. Take a small amount of that sucrose solution into the test tube. To that we add dilute sulfuric acid, about 5 to 10 milliliters of dilute sulfuric acid. That is heated for a little time using the water bath on the Bunsen burner, and then to that solution we add few drops of the blue color Benedict's solution, and then again we fix the test tube place it inside the water bath and we fix it to the stand and we allow it to get heated, and then you can see the observation clearly. So, when we carry out this first part of the activity, we did not get any color change. Therefore, we need to do the second part of the activity. In that, we prepare a fresh solution of sucrose and take that into a test tube. So, here take a small amount of the freshly prepared sucrose, that is sugar we use here, solution into a test tube. Then what do? We add the new reagent, that is sulfuric acid. Add a small amount of dilute sulfuric acid, that is H2SO4. Then heat the mixture. We don't need to heat it for a long time. Just we can place it in the water bath and take it out. After that, we add a small amount of Benedict's solution and heat the mixture using a water bath in order to get the proper observation. So the next step, add a small amount of Benedict's solution then heat the mixture using a water bath. So that is the second part of the activity. We need to heat the reaction mixture with Benedict solution after the addition of dilute sulfuric acid. And also I have mentioned before, in case we add an excess amount of dilute sulfuric acid, in order to neutralize that, we can add a small amount of sodium bicarbonate. That is to neutralize the dilute sulfuric acid if there is excess acid. So now you have understood all the steps involved in this activity. We have to do it in two parts. One directly with the Benedict solution, the other one after the addition of dilute sulfuric acid. So let us carry out this activity in the lab now. Okay students, now we will do the activity to test the presence of sucrose in food samples. For that, I will be using sugar, the normal sugar from the household kitchen, that is sucrose. And the reagent that we are going to use is Benedict solution. In addition to that, I explained to you all, we will be needing dilute sulfuric acid. First, I will prepare a solution of sucrose by taking a small amount of sugar into the boiling tube and adding little amount of water. As you know, students, sugar can dissolve very well in water. So, if I mix it nicely, you will get a proper solution. Now that the sugar has dissolved well and we have the sucrose solution ready. To this, I will have to add a small amount of the Benedict's solution. We have used it before also. You can see the color clearly. It is a blue color solution. So, I add a small amount of Benedict solution. When it is mixed, you can see it gives a nice blue color. Now, I used a boiling tube to make the sucrose solution because we have to heat it. For heating, we use a water bath. As you know already, water bath is needed because we need to observe the color change slowly and clearly. If we heat it directly, then the color change will be too fast. So, for water bath, we use a beaker containing water. I have already got it ready and you can see the burner as well as the tripod and the wire mesh. I will place the mixture into the beaker. You can observe the color change. We will have to wait for a little time. Students, you can recollect the color change that has to be observed here. As you can see, there is no change taking place. Still, you can see the blue color of the Benedict solution only. As you can see, students, even if we keep on heating this for a long time, there will be no color change because sucrose is a disaccharide. Then to obtain the color change, we will have to do the second part of the activity. So for that, I will again need a freshly prepared sucrose solution. I will remove this from the water bath and I can just place it in the test tube holder. We will just leave it aside. Now I am going to prepare another solution of sucrose. The sugar has to be taken into the boiling tube again. And when I add water, we get a freshly prepared solution of sucrose. Now that the sugar has completely dissolved in water, we will add a few drops of dilute sulfuric acid. Here you can see this is dilute sulfuric acid and I am going to add a few drops of it to the sucrose solution. And then I will be adding a small amount of Benedict's solution. Now we will heat the mixture using the water bath. Already I have prepared the water bath. We used it for the first part of the activity. Now observe closely. You will see the color change clearly. Try to recollect the color change, students. Now you can see the solution slightly turning from blue to green color. First it was a blue colored solution. Now it has become a green color solution. The color change will continue. You can see the solution changing color from light green to a yellow-green color. Now you all can see the yellow-green color becoming a little more darker. Now students, you can see the color changing from yellow to orange. If I mix the solution properly, the reaction will take place more uniformly. And now you can see the appearance of a brick red precipitate at the bottom. Although the solution is orangish in color, you can see a deposit at the bottom of the boiling tube. So this is the color change that you have to obtain. If there was glucose in the solution, the Benedict solution will change color from blue, then to green, then from there yellow-green, orange, and finally, like you see in this mixture, a brick red precipitate. So from this activity, when we added sulfuric acid, then Benedict solution and heated the mixture using a water bath, we got the color change. So from these, students, you can understand what has happened to the sucrose in the sugar. With the addition of sulfuric acid, sucrose has been hydrolyzed to glucose and fructose, and the glucose gave the color change with Benedict solution. So, you have seen the observation clearly and you have understood why we did this activity in two parts. First, there was no color change, then we saw the color change very clearly. There is one more step that you can add to this activity. In case you have added an excess amount of dilute sulfuric acid before in the previous step, then we can add sodium bicarbonate to neutralize that acid, and thereafter we can add the Benedict solution and continue the activity. So, that is an additional step that you might have to remember. Okay students, this confirms the presence of sucrose in sugar. Okay students, you observed the activity clearly and you saw the color change that took place at the end of the activity. So, we did this activity in two parts. First, when we heated the sugar solution, that is the sucrose solution with Benedict solution, what was the observation? There was no color change. That was the first part of the activity. So, I will write that first. No color change when the sucrose is heated with Benedict's solution. So, what did we do? We prepared another solution of sucrose, added diluted sulfuric acid, and then after heating, we added Benedict solution. Then when we heated the mixture using a water bath, we got the proper observation. And you saw the color change from blue to the formation of brick red precipitate. So, after the addition of dilute sulfuric acid, what was the color change? Initially, the Benedict solution is blue in color. So, the reaction mixture was blue in color. So, first the mixture was blue in color. Then this blue color became green. And the green color became green yellow. And the yellow color became orange. And finally, we got the brick red precipitate. So, you can remember these observations clearly, students. Blue, green, green yellow, orange, and then brick red. That is the color change that you get when you do this reaction using sulfuric acid and then adding Benedict solution. So, why does this happen? First, we heated the sugar solution with Benedict solution. There was no change. That is because sucrose is a disaccharide and the Benedict's test gives a color change only with monosaccharides. monosaccharides. Then when we heated the sucrose solution with dilute sulfuric acid, you know the process. There is hydrolysis taking place. So the sucrose was hydrolyzed to glucose and fructose, and the glucose gave the answer to the Benedict's test. So for a Benedict's test to give a color change, there should be glucose in the mixture, or there should be a monosaccharide in the mixture. If it is a disaccharide, when you heat it with dilute sulfuric acid, it undergoes hydrolysis, and the hydrolyzed product produces glucose. Therefore, it gives the color change. So to conclude, we can say that in the first step, sucrose does not give color change with Benedict's solution. But when heated with sulfuric acid, with H2SO4, that is sulfuric acid, what happens to the sucrose molecule? It gets hydrolyzed. Sucrose gets hydrolyzed to glucose and fructose. Then what happens? Glucose gives color change with Benedict's solution. Gives color change with Benedict's solution. So that is the reason why we did not get a color change in the first part of the activity, just heating sucrose with Benedict's solution. Because it was a disaccharide, it did not give the color change. But on heating with sulfuric acid, it gets hydrolyzed to glucose and fructose, and the glucose answers the Benedict's test. So students, now you have understood the activity. You saw the activity being carried out in the lab and you observed the color changes. And now you know the reason why we don't get the color change in the first step, but we get the color change in the second part of the activity. So that is the activity to test the presence of sucrose in a food sample. Now students, you have understood the different types of carbohydrates, the elemental composition, their basic structure, and the different examples, their significance to living organisms. So that is the first type of biomolecule that is present in living organisms. We have already discussed there are 4 types of biomolecules. Now we will discuss the second type of biomolecule, that is protein.